Assalamu alaikum wa rahmatullahi wa barakatuh. Good day, everyone. This program is part of the National Science and Mathematics Olympiad, NSMO, that aim to develop scientific skills. We will focus on analytical thinking, problem solving, and scientific exploration. Welcome back. Biology in this phase focuses on understanding life from the cell to the entire organism. We'll learn how cells function, divided, and why this is essential for growth and repair. We're still in chapter 4, and we are continuing studying one of the most essential biological processes, cell division. Without cell division, there would be no growth, repair, or replacement of damaged cells. So previously in this chapter, we studied Introduction to Cell Division, and we done with mitosis. And now we will take regulation of the cell cycle. Okay, and these processes create new cells and maintain chromosomes number. This lecture explains how cells regulate the process of division. Regulation prevents errors and complications such as cancerous growth. We will explore cell cycle control, errors leading to cancer, and stem cells. These concepts are key to modern medicine and future research. The best way to understand this concept, regulation of the cell cycle, is to answering the serial following questions. So, the first question, why does the cell need strict regulation of its cycle? Because uncontrolled division may lead to abnormal growth such as cancer. So here probably we have cancer instead. So if we don't have a control, probably we have something called cancer, abnormal of growth. Second question, what controls the movement from one phase to another in the cell cycle? It's controlled by proteins called cyclin. Here, cyclin. Who's the controller? Cyclins. So, and cyclins and plus kinase enzyme. Kinase enzyme. Both of them would control cell cycle. Okay. Let me put check for 2. 3. How do cyclins work in this regulation? They rise and fall depending on the phase, allowing transition at the correct time. So they depend about be on or off, on, off, about the phase they dealing with. Okay, so this is the question number 3. Now let's be focused in some spot in the figure in the right. First one, G1 is checkpoint here. Let's discuss these checkpoints. And this checkpoint raises this question. What does the cell check before entering its phase? It checks cell size. Let me write it here. Cell size plus nutrition plus sufficient and other factors, which is growth factors. Growth factor. So that one, it would be checked before going to sDNA. That here, sDNA. Okay. Other question. In spot of G2 M checkpoint, M it's referred to mitosis here. Let's have the first question. What must be completed before mitosis begins? It must check DNA replication error to be sure there's an error free. Here, the most important is to check there's no error. or error free. Okay, this is question number 5. Okay, going with another spot, which is spindle checkpoint. Here, spindle checkpoint. Why does the cell pause during metaphase? Why? To ensure all chromosomes are probably attached to spindle fibers. So here we want to make sure all chromosomes attached with chromosome. Okay, so all spindle fibers attach chromosome. Last question, what happens if checkpoints fail to work? What would be happened? Probably the cell may continue dividing uncontrollably, and if the cell dividing uncontrollably, probably leading to what we called it, and you know it, cancer. Probably lead to cancer. Imagine that, guys. The cell cycle works like an airport. The cell cycle works like an airport. No plane takes off without clearance. Another very important terms on regulation of cell cycle, they called it apoptosis, is a programmed self-destruction of damaged cell. So this is a process that occurs in certain cells, leading to death of cells produced from those division. And also it protects the body from abnormal cancer cells. Let's explain the figure here. We start with the first one here, signal. So the cell receives signal telling it that it is time for a programmed death. This is the first one. Second, cell shrinkage. The cell begins to shrink and lose its normal stage. Number 3, formation for apoptotic bodies. This is apoptotic body. The cell breaks into small bubbles, like pieces called apoptotic bodies. Number 4, nuclear fragmentation. So the nuclear membrane breaks and the DNA fragments into small pieces. And that leads to number 5, which is removal. Other cells like white blood cells. Imagine that white blood cells like this. Okay. Remove these pieces safely without inflammation. Okay, so apoptosis happen quietly and orderly, like demolishing a building without causing damage. Let's have 2 examples of apoptosis in a human and in a plant. In a human, during development, cells between fingers, that was here before, die to shape the hand. In plants, leaves fall in autumn due to programmed cell death. And also these probably happen in more of living organisms. All of us need cell to be divided to save our lives, but with certain of time. We know cell divided probably from 1 to 2, 2 to 4, 2 to 8, but it will be divided in a certain time. If the cells will not hold these terms of be divided in certain times, it will be causing for a problem, abnormal growth, that names really known as cancer. So cancer is uncontrolled and irregular growth of cells. It occurs to DNA mutations or environmental factors like smoking and radiation. It may be inherited when mutations are passed through generation. Okay, let's see the picture here. This image shows a cluster of cells dividing rapidly and uncontrollably, representing tumor growth. The second image here represents environmental factors that damage DNA, such as smoking, UV radiation, and asbestos. Picture number 3 here shows mutation or changes in DNA strains, the root cause of cancer formation. Last picture here symbolizes that certain type of cancer can be inherited genetically from parents to children. Okay, guys? So, cancer starts from a mutation in DNA caused by environment or inheritance. Then the cell grow uncontrollably. Let's talk about another very important term, which is stem cells. So, stem cells is undifferentiated cells that can develop into various cells type. Embryonic stem cells are more potent than adult stem cells. Okay, they are used for treating paralysis and organ disorders. Let's have for sure there's two types of stem cells: embryonic stem cells and adult stem cells. The differentiation between embryonic stem cells and adult stem cells is embryonic stem cells have a higher ability to specialize into many types of cells. On the other hand, adult stem cells are found in the body after birth and can produce only limited types of cells. All in all, stem cells are the body's backup system, ready to turn into new cell when repair is needed. So we learned today about cell cycle regulation, cancer, and stem cells. This knowledge is fundamental to understanding modern therapeutic medicine. Thank you for your time. Thank you so much. See you soon. So long.